Thanks for that introduction, and thanks to everybody for coming. Let me get my click on the right spot here. So I would like to, have us think a little bit about using our classroom response systems. Quite a few of our faculty use clickers, and we try to use those to engage our students in different ways. But just talking with different faculty within general biology and within the broader division of biology, because that's where in the division of biology, that's where we have faculty that are in the microbiology, the biochemistry, cell molecular biology, and ecology and evolutionary biology departments. People are always trying to think about kind of creative but yet easy to manage ways to engage students and come up with in some cases you're looking for some like abilities for students to get some points for attending, but you don't want it just to be attendance points. You're trying to actually engage with the students. And yet sometimes people will struggle a little bit with using clickers in the classrooms because you may kind of fall back to just asking really, really simple multiple choice type questions, and you feel like that you're not really engaging the students as much as you would like. And so just as a reminder for anybody if there's anybody here that's not really familiar with clickers, clickers started out as actual physical devices. And there's been several generations of those. Actually, I think the first clicker that I ever used years ago was a different brand than these. And it was kind of like a TV remote, and it just had enough buttons where you could vote A, B, C, D, E, or enter 1, 2, 3, 4, 5, or something like that. All you could do was multiple choice kinds of questions. And the various vendors that are producing these classroom response systems now then went through a period where they came up with some devices that look kind of like an old BlackBerry with a small keyboard, so that you could enter in short word or even phrases, and those in a certain scenario could be projected up on the board when you get the collective responses from the students. Currently, UT is using Point Solutions as the app, and it's a little confusing because the company is merged with Echo 360, and sometimes things are listed as Echo 360, sometimes Point Solutions, but the app is Point Solutions. And the truth is, a lot of students, I know in my class, when I'm in person, the students are welcome to use their smartphones or their laptops so that they don't have to buy a device like this. The class that I'm teaching this semester is fully online, and so they have to be using a mobile device of some sort. But still, I can receive their responses and they can get points for those responses. But the challenge is, how can we actually engage our students? And you're not just asking students, are you here? Are you not here? And they get some points, or you're asking just really simplistic kinds of questions. And so I'm going to give you just a few examples here, and feel free if people want to break in and ask a question. You can unmute yourself even if you have a question about things. I'm just going to give you a few examples of some kind of case study like questions that I use. And then I'm going to get to kind of showing, which is kind of the point of this presentation, what I think you may find to be a little bit of a different way of utilizing clickers, structuring a different type of question, different kind of scenario-based questions that you may be able to adapt with some of the classes that you teach. And so I kind of like to offer scenarios in my Bio 101 and 102 classes, and I'm trying to do this in my the new Bio 104 class. And this is an example of a question that I've asked in 102 before, very early in the semester. So the students don't have a lot of background as to what's going on in this particular scenario, but you could ask initially, let them know these are horseshoe crabs. Some of the students don't know what a horseshoe crab is, but these are horseshoe crabs. And if I was in person, for instance, I could be asking the students to try to pick out where's the female. Actually, if y'all would be free to enter into the chat if you want to, and kind of guess in that little upper image, which one's the female? Is there some way that you could tell which of those particular horseshoe crabs might be the female? Okay, Katie's jumping in. She's voting for the middle. Okay, other people, yeah, give you a quick warning. Somebody else is going for the middle. And so as you're like typing in, I'm gonna go ahead and jump in here. It's like, indeed, it's the horseshoe crab that's in the middle. The ones that are on the outside, those are the boys. And so truth is, horseshoe crabs aren't that much different than college fraternity brothers, where the boys are running around. It's like, please have sex with me. Please have sex with me. You know, it's there. We haven't evolved all that much. That the boys are chasing the girls around, desperate to have sex with her. And that's what's going on all up and down the beach right here. There are thousands and thousands of horseshoe crabs that are all breeding at a similar time of the year. And so then I would have the students think about, well, why are they breeding at the same time? What might be the advantage of this? And again, this is very early in the semester. We're not really talking about, we haven't talked about competition. We haven't really talked about reproductive strategies. I'm throwing these kinds of questions at the students in a sense to gauge where they are, what their life experiences are. Do they have some misconceptions about something? And then we can just kind of engage them where they are and go from there. And so for here, you can see that the students are kind of split between C and D. In reality, both of these are correct. And actually, I kind of like to have questions like that are sometimes in the clicker questions that I ask were in reality. Biology is not always simple. Sometimes we oversimplify biological kinds of problems, and then the students are expecting everything to have just a really simple, cut and dried answer. But the reality is sometimes it's complicated. And so in this case, what's going on is it boosts the rate of fertilization, because once the females release their eggs, in truth, there are going to be several males that are in the area that are releasing their sperm into the water. It's going to increase the rate of fertilization. But then having so many eggs fertilized at the same time increases the likelihood that at least a few of the young might somehow survive. The reality is that the survival rate of species like horseshoe crabs is shockingly low. Most of the eggs end up getting eaten by birds and fish and other things. It's really only, so you need to just overwhelm the system with lots and lots of fertilized eggs before you're going to have the chance of really hardly any of those young, those larvae, developing to adulthood and themselves being able to reproduce. Here's another example of that. Here's a photo that I took when I was going down to Tampa Bay for a workshop, and I was sitting at the window seat and looking out over Tampa Bay. There's the kind of greenish tint areas of the water are representative of some areas that have an unhealthy level of algae and photosynthetic bacteria that are sometimes referred to as cyanobacteria. That's an algae bloom, and that's not particularly healthy. Where does that come from? And so, again, you can sometimes ask these questions where you might have talked about some aspect of the concept in the past, and you're hoping that the students kind of connect the dots and get to where you're wanting them to go in that particular series of questions. And in this case, it's all about the nutrient level in the water. Most people don't really realize that a healthy aquatic system doesn't have much phosphorus and nitrogen in the water that's in a chemical form that's available for photosynthetic organisms. And the more nitrogen and phosphorus that you have in the water, the more likely you're going to have an algae bloom. And we can use that then as the launching off point in class to talk about, well, what's the downside of having an algae bloom? Why is an algae bloom a bad thing? It's always nice to have a scenario where the students are making a connection to the local area and even the campus. And so this is actually, no kidding, the largest recorded, documented willow oak in the state of Tennessee. This is a state champion willow oak that's across the street from Strong Hall, where I am, over by Henson Hall. I'm really concerned with the expansion of the business buildings that are going to be going over there once they tear down the buildings in that area and start building the new business building. I'm hoping I don't have to chain myself to that tree, because I don't want to lose that state champion willow oak. But this was has been a jumping off point, and I don't have the student responses in this particular question. But you could ask the really tough kind of question, and lots of people, even biologists, struggle with this. What's the source of the majority of the dry mass of a tree? And, is it nutrients from the soil, nutrients from the air, nutrients from the original seed, water from the soil, or something else? This is something that, no kidding, botanists took forever trying to figure out. Where does the mass of a tree come from? And the reality is it's nutrients from the air. It's mostly the CO2. That's most of the dry mass of the tree. And so just using questions like this, scenarios that the students can potentially relate to a little bit, and then use that as a jumping off point to start to investigate more complicated kinds of topics. Let's see, I'm gonna skip past that one right now. Here's really the kind of focus of the style of question that I'd like for you to maybe consider trying to adapt for using in some of your classes. I've actually used this scenario the very first day in Biology 102. And I'm going to show you some of the student responses from, this is the very first day in Biology 102. This is mostly non-science majors in this class. So let's see what you think about this. Okay, so these structures that you can see from the air are things that are referred to as fairy circles. And so these are in Africa. I'm going to challenge your geography here just a little bit. It's in the Namib Desert in Namibia. And biologists and geologists have been fascinated by these things. Here's another couple of pictures to give you a little better sense of the size of them. Depending on the time of the year, the grass is either really stunted and dried back at the dry part of the year, but when there's been some rain, you can tell there's a different type of grass that seems to be circling that dirt that's in the middle of those fairy circles. What causes these things? Okay, now here, I want you to put into the chat, if you would. What do you think contributes to or causes fairy circles? And so if I was in an in-person class, I would project a question that looks something like this. So you can see there's nothing for A, B, C, D, E. I would actually walk around the classroom and try to encourage groups of students to come up with some ideas or suggestions about what they think is contributing to this particular phenomenon. I'd write those on the whiteboard, and then the students can vote A, B, C, D, E, and they can see their responses. Now, I'm sitting here. I've already put all y'all to sleep. Apparently, no one is offering. Okay, aliens, there you go. Aliens, fungi, getting a few. Availability of water, okay, water source. Parasites, that's an interesting one. Okay, so these kinds now I've actually found, and I've done a number of these in my Bio 104 class where my lecture is synchronous, so it's live. And so I can just have the student, if you've got 168 students that are in a class like that, and you ask them to put stuff in the chat, oh boy, the chat just completely blows up. And it's like, it's hard to keep up with all of the things that the kids will put into chat. They won't talk to you in a in-person class 9 times out of 10, but they will chat up a storm. And so then I'll kind of go out of the full screen display mode, type in their answers into the questions, and then go back to full display and let them vote on it. And so then they're actually having to vote on their suggestions, their proposals, their sometimes even rudimentary hypotheses about what is going on in that particular scenario. And so I'm going to jump down here. This is actually from spring 2021, because I went back and I found a slide where I hadn't cleared it and I still had the percentages from where they had offered some of their suggestions. And look at that. A few of these are similar or in the general area of some of the suggestions that you guys are suggesting. Nesting birds, potentially, they're making that kind of clear area. Chemicals in the ground, bad soil, not enough water. So couple of you had something about water availability, insects. There's actually one of the things that the local tribes, the way they describe these fairy circles is that these are access points for the spirits of their ancestors coming up and out of the ground. So it's like they're visiting the afterlife, which is underground, or that there are spirits that are coming up and out of the ground, and that's part of the local legends that would describe where these fairy circles come from. And so in truth, geologists went out there and they're like, you know what, this is kind of a geologically active area. Let's see, are there like volcanic gases or, sulfur containing compounds that are leaching out of the ground at different points? Is that contributing to it? Here we've got primarily freshmen are describing some of the things that professional scientists initially went to trying to explain why these fairy circles might appear. And as it turns out, there's no chemicals that seem to be coming out of the ground. You take soil samples from in the middle of the circles, outside the circles, and nobody could find variations in the chemical composition in different places. When it rains, the water goes everywhere. So why would it pool? What would keep it in one area versus another? We'll kind of come back to that in a second. One of the primary hypotheses that people were putting out at one point is that there might be nests of termites in the center of those fairy circles. And so then in class, we could start talking about, well, if you think that it might be some kind of like ant or insect colonies that are causing these things, how do you separate correlation versus causation? So you might be able to look in a fairy circle and say, yeah, I've tested a few of the fairy circles and it seems like that there are termite nests, but which came first? Were the termites there and that caused the circle, or did they move into the circle after it was already there? And so then we're actually on the first day of class jumping off and starting to talk about the difference between correlation and causation, and what kind of information would you need to actually generate a experiment that could be tested to find out if your particular hypothesis might be valid or not. And this back in spring of 2021, actually again in spring of 2022, because some of them came up with a list that was similar to this, pointed out that in fact there are termite nests in many of these fairy circles. It's a underground type of colony. It's not like some types of termites build these great big above ground nest, and the ones that are in Namibia, in this particular area, they have a colony that's completely underground. And so you actually have to go to the fairy circle and dig it up and try to see, is there a nest here? But the students, after we discussed it for a few minutes, they realized that what you really need to do is dig in the middle of a bunch of these fairy circles and make sure that there are termites in every single one of them. And then some of the students actually suggested the possibility that could you find the queen and some of the workers and transplant a termite colony to a new place where there's not a fairy circle, and can you form a fairy circle? Because now you have a nest in a new location. So they were actually coming up with some interesting suggestions. Now, what's really causing the fairy circles? As it turns out, there are some fairy circles that don't have termite mounds, which has undercut this idea that the termites are causing the circles. And I think one of the more possible hypotheses that seem to be gaining some strength now is that there's a different population of soil fungi in this area that helps retain the water in the middle, and that is in part what contributes to the initial formation of the fairy circles. And then here come the termites, and they're grazing on different seeds of different species of grasses, and that might contribute to maintaining the fairy circles, or kind of refining the edge of it. So bottom line, we still don't entirely know. There's still things to test, which again is an important message for students early in a biology class, where it's not just about memorizing correct answers. Science is a process, and sometimes you get some of the information, but you haven't quite addressed the question. And so here's really quick, let's look at one more example that I've used. So this is Slinky. This is a fish that used to be in my son's fish tank. And every now and then, the tank would get overgrown with algae. Guess what? That's tying back to our algae bloom question about Tampa Bay earlier. So this is one of these questions that you could have one and then the other, maybe a different day of class, and see if they make the connection there. But I could ask the students, okay, what happened to poor Slinky's tank? And give them the open-ended clicker questions. And so they can either offer suggestions in an in-person class, or they can put things in the chat. I'll type those in, and they get to vote on their suggestions about Slinky's tank. And what are some of the things that they have come up with in a previous class? They came up with Slinky's bringing out CO2 that's fueling photosynthesis. Maybe it's waste from Slinky. It's too much sunlight, too much heat. Somebody came up with nitrogen discharge from Slinky, which of course that's really what's going on with B. But whatever they come up with, I put that into the questions, and the students get to vote on it. And then we get to talk about it. Now, these kinds of questions I've found have been quite useful in getting the students to think about and actually make some predictions and engage in some of the material. And then you've got a good starting point for a conversation with the students after that. Now, what am I doing with these? When I first started doing these kinds of questions several years ago, I just wanted to come up with some way to get the students to kind of think about the material, be able to vote in a format that I could easily upload to Canvas, because in my Bio 101 and 102 classes, so these are classes with like 220, 230 students, I don't have a TA in the lecture class, so I can't really be picking up assignments and having those graded. I don't really have grading assistance for the lecture part. I have to be able to just upload the results from the student responses in class somehow, and I was trying to use the clickers to do that. Well, I actually presented, we had kind of a general biology retreat workshop this last academic year, and I presented that fairy circle example with a small group of people in our group. And one of my colleagues, Meryl Roseberry, I'll shout out to Meryl, looked at that and she's like, that's the 5 E's. I'm like, that sounds familiar. Remind me what that is. It's basically the 5 E's of inquiry-based learning, where you look for a mechanism where you can engage the students in a particular situation, give them a little bit of information, provoke some questions, let them explore this either individually or as a group. They can come back and explain potentially the phenomenon. You can help elaborate what's going on in that particular scenario, and then you can have some follow-up questions, and the students, you're able to allow the students to evaluate that scenario in a bit more depth. And so there's actually a solid pedagogical basis of doing questions like this. And I can report that this actually has worked really well, both with in-person and online classes. And since Meryl recognized that this is what I was doing in the lecture class, she's now our 101, 102 lab coordinator. She's working more closely with the TAs in those courses. She's like, we ought to do this for the TA meetings. And so sure enough, we've been over this last year trying to incorporate more questions, scenarios like this in PowerPoints draft PowerPoints that we'll give out to the TAs. And we have the TAs practice going through these kinds of scenarios in the weekly TA meetings. And that allows the TAs to kind of develop a little bit as to how can I encourage my students in the in-person labs to engage in the whatever the lab material is for that particular week, to start asking questions, start conversing with the other students that are in the classroom. And that, I think, has started to increase a little bit as the semester goes on, the comfort level of some of our TAs interacting with our undergrads and those 101 and 102 labs. Because after all, a lot of times we get first semester, first year grad students largely in a class like 101. And so the TAs themselves are going through kind of a tough learning curve of how can I manage a classroom. And so this is actually turning out to be an interesting strategy to help engage the students in at least some of these interactions with the students in these labs. Igor, I don't know, where are we with time? Do we need to, I've got another example that we could go through, or we could go ahead and
probably about a few more minutes so we can allow the audience to ask questions, and then we can also explore use cases for asynchronous delivery.
Okay, so I'm gonna just,
okay?
yeah, I'm just one more example at you.
Okay, thanks.
And so this is a Tasmanian devil, and so it's not the cartoon. Usually, if I was in an in-person class, I might, or even in a synchronous online class, I might show a little video so that they can see just how aggressive. They're kind of scary. They're kind of cute, but they're kind of scary. They'll really go after the zoo handlers. And the next slide is not so cute. The Tasmanian devils are at risk of what has come to be known as the Tasmanian devil facial tumor disease. And so what happens is that these things, the Tasmanian devils show up with extensive tumors around primarily the face, the mouth, around the eyes, And this cannot be easily treated because these are wild animals, and it's spreading through the island of Tasmania. So if you're, again, challenging your geography here, the island of Tasmania is south of the continent of Australia, and the Tasmanian facial tumor disease was first identified in the eastern part of Tasmania, and it has spread to the west and to the south. There have actually been wildlife managers that have proposed that one of the possible ways to save the Tasmanian devils is to maybe go to some of the places where you don't have the facial disease yet, collect some of those animals, put them in zoos and reserves, and just let the disease burn through the countryside and kill everybody, and then reintroduce some of the captive ones. And so this is like a really difficult scenario. What do you do? How can you track this disease? And so one of the questions that I would ask is, what might be the source or cause of the facial tumor disease? And so once again, I would have the class. We're not going to make y'all do this right now, but they put stuff in the chat or offer suggestions in person. And I don't have the student responses from it, but this is the list from a previous question or from a previous semester, where some of them were suggesting that it might be a virus that's causing the disease. It might be local pollution, it's something in the food. Maybe there's something chemical in fertilizer that's in the area that's causing the cancer, or something else. And I'll just point out that I'm using this particular scenario as an introduction to the immune system. So we've just talked about cancer, and we actually had a few examples of cancers that can be caused by a virus. I mean, in humans, cervical cancer is primarily the result of the human papillomavirus. And so we had talked about that, and it was good to see that quite a few of the students suggested that maybe this is caused by a virus as well. And in truth, biologists, that's one of the first things that they started looking at, tried to capture some of these Tasmanian devils, get some of the tumor tissue, grind them up, look for viruses that are associated with the tumor, but not healthy Tasmanian devils. And bottom line, it's not a virus. So what is it? As it turns out, this is a transplantable tumor. There's only a couple of examples of this in other species. As it turns out, the primary tumor, we now know there's actually two. It's a little bit more complicated than just a simple single type of cancer. There's actually at least two types of cancer that have formed over the years. The one that's most widely spread is actually derived from a nerve tissue in a female Tasmanian devil. And as it turns out, that tissue does not express the proteins on the surface of the cells that your immune system ordinarily would be using to discriminate, is that tissue me? Is it self, or is it non-self? And so those cells don't let the immune system know that that tissue doesn't belong in that animal. And so those animals cannot launch any kind of a tissue rejection or an immune response against that tumor. And so the Tasmanian devils accept that tumor as being their own tissue, and it ends up killing most of them. And so it's a real struggle now. As it turns out, there's some evolutionary genetic evidence that the Tasmanian devils may be starting to develop resistance against one type of the tumor, but there's a second type that is actually developing mutations faster than the other. And so it's still up in the air whether the Tasmanian devils can be saved as a population. But this became then a example of thinking about tissue discrimination of the immune system, self versus non-self. And so these kinds of questions can be utilized for a variety, I think, of different purposes, tying together information from different parts of a class, lecture, potentially labs and professional development for TAs. And then part of the question would be, do some of you have some ideas or questions about how you might be able to use these kinds of questions, I think, in your classes? And so I'll stop with that. That's my break.